Hello, I'm Ben Knapp and I'm the superintendent at the Basket Wildlife Research and Education Center. And in this video, as part of the virtual field day, we're going to talk about a study where we're looking at legacy effects of agricultural land use on forest development. As we've seen in the intro video, the Basket Center historically was a series of farms that were brought together and now are University of Missouri property. The legacies of that agricultural land use on those farms has left an important mark on what we see in terms of forests today. So for this study, we looked at the 1939 aerial photograph to look at places that were previously cleared of forest, so they were either pasture or cropland and had no trees or very few trees. And then we also looked at places that had forest in the 1939 photo. So at that time they may have been grazed, they were still being used by people, but they did have forests that were intact at that point in time. And this created two different starting points from the 1930s through today. So as part of this study, we're interested in how those agricultural legacies created different forest structures and compositions compared to what we see and what was forested at that time. Now we also wanted to look at how soil type may influence some of those patterns. So within the same area, we use soil survey maps to look at some common soils across those two different conditions. One soil is within the Weller soil series, and this occurred more on flat areas and had a fairly deep lust cap, and this was pretty heavily used for agriculture. Now off of those flat areas onto slopes, we got more into the Bardley-Klinkenbeard complex, which is a stonier soil and tends to have more sloping features. Because of the abundance of these two soil types with the two legacies, agriculture and forest, we're able to create combinations of these where then we could sample and look at how their combinations affected what we see today. So we installed 6 sampling plots within each of these 4 conditions so that we can get an idea of what the forests look like today. Now we're standing in one of the agriculture legacy plots on the Weller soil. So this was a condition in the 30s that had been cleared of forests and used either for cropland or pasture. Over the past couple years, we've worked with a number of students, often as student interns, to collect data on these plots. So this particular plot, we've sampled the overstory trees. So we have a tree tag on this cedar behind us. We've looked at the mid-story development, so some of the smaller trees, smaller diameters that occupy the mid-story class. And we've also looked at the plants that are on the surface, which include a lot of invasive species and some of the herbaceous plants as well. Through the sampling and what we've learned, a plot like this has a lot of eastern red cedar that's moved in through time and often is dominant within the canopy. There may be other species mixed in. A lot of times these species would be trees that we associate with disturbance or early succession. So perhaps black locust, elms, hackberry. We also will see some maples, and we see some oaks, more in the red oak group within areas like this. Within the mid-story and the understory, we have a lot of invasive plants. And so species such as autumn olive and honeysuckle, as well as Asian bittersweet and multiflora rose, are very common in this type of condition. You can learn more about those species and other invasives in another video that we have within the virtual field day. Another aspect of this project was setting out camera traps where we could look at wildlife use in these different conditions. And we had an undergrad student, work on that aspect of the project. Through that sampling, we collected thousands of photographs of animals as they used the different habitats. We found some interesting results based on those camera trap photos. Now, a more formal analysis is ongoing, but just based on the pictures and the abundance of species, we found that species like rabbits and some small mammals preferred or were more abundant in these habitats, likely due to the presence of cover for them to be able to hide in. So now we're in a forest plot, and we can see here that the structure is very different in terms of the way that this forest looks. So we have a few very large trees that make up the overstory, and most of these are oaks. So we have white oaks as well as some red oak. Here's a northern red oak here behind me. And so we've got this layer of large canopy trees that are pretty old. They may be 100 years old. One component of the study we're still working on is doing some aging so we can reconstruct how these forests came together in terms of their structure and composition. In the mid-story of these plots, we have a lot of sugar maple. And that's most of the stems that we see that are 1 inch to 4 inches in diameter. Most of that is sugar maple. Now as we look at the forest floor, we see a very different composition. So there are not near as many invasives out here. The abundance is much lower, but we still have some of the same species. We still see autumn olive. There is some Asian bittersweet out here as well, but just the numbers are so much lower than what we see in the ag legacy plots. We have a notable lack of oak seedlings on the forest floor here, and most of the tree species we see are ash or dogwoods, but we don't have a lot of the oak underneath. Now, based on the camera trap work that we did, we found that deer and turkey use these habitats much more than what we saw in the agriculture legacy plots. A final aspect of the study we're looking at is the soils themselves and how some of these legacy conditions have affected soil properties. So recently we've done some soil sampling to look at various characteristics of the soils. And next we're going to have a student who worked on that project, Sarah Higgins, talk about some of her research.
Hi, my name is Sarah Higgins. I'm a student here at the University of Missouri in Columbia. I am in the School of Natural Resources. I'm entering my senior year, and I'm studying environmental science with an emphasis in soils. Last summer in 2019, I did a research project here as an undergraduate here at Basket, looking at soil health parameters in areas with different historic land uses that are currently forested and comparing them on different soil types to see how they changed. While I was working on this undergrad research project, at the end I produced a poster to show these findings, which you'll be able to see. So with this project, our objective was mainly to look at soil health through measuring PLFAs, or phospholipid fatty acid analysis, to get a read on the biology occurring in these soils at different depths and down the profile. We also looked at total organic carbon and fungi and actinobacteria. So we sent those samples to the Mizzou Soil Health Assessment Center where they were analyzed. So we had 12 plots we looked at, and they were all restoration plots here. They had 2 different types of soils mapped, Wellers and Bardleys, which you'll see here on this tour. So we had 3 plots for each mix of the land use. So we had 3 Wellers in a forest, 3 Wellers on a historically agricultural site, and then 3 Bardleys in a forested historic use site, and 3 Bardleys in a historic agricultural use site. So the data we got back from that was really fascinating. We found that regardless of land use history, the Bardley soil type had more PLFAs, and that's just a read of biological activity, so more biology, soil biology, inherently than the Weller. And we also found that on the profile level, so the whole pit, the Bardley, the forested, historic forest land use had more soil biological activity than the agricultural historic land use. And however, in just the surface, so the 5 centimeters, the agricultural land use had more soil biological activity than the Bardley. Another really cool finding we found was the really strong correlation between total organic carbon in these soils and predicting the amount of PLFA that's also in the soils. So in regards to soil health, a lot of what you're finding can be determined by how much total organic carbon you have, and that could explain some of the other findings we found. So this is really cool information to enhance other research projects going on out here that can be used, and also can be used in ecological restoration to help know what's going on in the soil. Hope you enjoy your virtual tour of Basket. I enjoyed working here, so have fun diving into these soils.